So thank you very much for for hosting me, and thank you everyone for joining. And I believe that this is going to be an interesting moment for us to talk about things that are very common to us and that also relates to ourselves and our health. Um, so I want to begin by saying that we know that human health, animal health, and even the environment we are, I mean, there's an interconnectedness between every living thing. And even if you are looking at it from the biological, ecological standpoint, you will consider food web, food chain, and all those kind of ecosystem network. So in this sense, anything that affects one of the trophic level affects the other one. I just want to come from that direction. Um, for example, if a tertiary predator like the raptors, anything happens to them, um, the prey and all the way down to the very basic level of the trophic level will also be affected. So in that sense, there needs to be a balance in nature. And that is what has been missing for a very long time until the the one health approach was introduced, where the health of the environment, the health of people, and the health of animals are considered to be very, very important. But then ultimately, if the environment and animals and people are kept healthy, then the people actually do the much of the benefits in the sense that everything is pointing to the fact that human being wants to live healthy. So in that case, they want to keep the environment healthy. They also want to keep biodiversity healthy, in which the biodiversity plays a very, very key aspect in maintaining and distributing diseases that affects humans. So I think the people came to their senses to know that okay, if we conserve biodiversity, we are also saving our health. All right. So and that is actually the beginning of the the presentation, which says conserving biodiversity and monitoring wildlife health are not actually just ecological goals, but the ultimate goal is that we have human to be in good health. All right. So, what is this One Health approach? Um, One Health, as I've briefly explained, is just about having the environment, the people, and everything that are interconnected to be at equilibrium, where there will not be some kind of a shift to a particular direction, thereby affecting other aspects of the system. For example, in an ecosystem where you have mosquitoes, ticks, birds, human beings, and all animals are interacting, one disease can really affect the other population of other species. For example, if a mosquito carries a virus, it bites humans, and that becomes something very, very important to consider in the sense that it leads to sickness for humans, and it's like a cycle where if mosquitoes are infected, they bite the birds get infected, the birds come close to humans and even domestic animals, and that interaction creates an interface for more um, transmission to go on. So it is this One Health approach has been designed in a way that it's not just only the medical doctors that should be doing the work of human safety, and it's not just only the Dr. Panton. Sorry, I'm not. I'm not saying that's been biased towards you. And then it's not just the veterinary doctor that should be in the circle, but the ecologist, as well as the sociologist, everybody should come together to make human health a surety. And so in that case, we monitor they monitor animals, they monitor the environment to make sure that the health of all these systems are kept in equilibrium. So where does conservation really fit into this One Health approach? There was a time back in those days, really like over 10 years ago now, I guess, maybe like 7 years or 8 years. So I was asked in a class by one teacher, and then she asked, if we clear the forest and the animals in the forest. So we're talking about disease transmission in this situation. And then she asked, if we clear the forest, where do the animals go? And what happens to the diseases? And then it was a very, very silly answer I gave to to her. And I say, if you clear the forest, the animals will run into the bush more and more. So, so it um, thereby it will just take them away, and we're not able to. It reduces the risk of disease transmission. But in reality, that is actually the opposite. And I can stand all the days of my life that I was wrong in that answer. The more we clear the environments, the animals don't have enough place to stay, and then they come towards the place that we have cleared, where we have built our house, and then they come all the way to the urban areas, and then there's this kind of complex interaction that is happening between humans and animals. And that creates an interface for a high risk area where diseases can easily be transmitted from animals, from these animals to human. Take example, a look at this picture of a bat. I'm talking about a bird, but I just feel that this picture is a very perfect example of talking about how conservation fits into the One Health. If humans will not affect or will not go to hunt bats. This is actually an adolon, the straw-colored fruit bat. Uh, yeah, people think that they carry coronavirus, they carry um, hantavirus and all of that. So for example, if this bat is carrying these viruses, all right. And this person goes to hunt this bat, kill it, and skins it. Skins it this way in a very, very unhygienic manner with your hands, bare hands. You can imagine what this person is putting himself into. So it's a very, very high risk, you know, is there dressing it and all of that. And it's not that he has roasted this bat. He just removed the skin without roasting before he boils it. So it's really a matter of concern. So animals are reservoirs of diseases. So if we let them be, we will not worry so much about contracting these diseases, because if we disturb them, they get back to us. They come to our places, they come to our compounds, they come and then we interact with them, and then disease is spread. So the best means to do this is actually to avoid habitat disturbance and to avoid hunting. And as well, when we avoid this habitat disturbance and hunting, biodiversity will thrive. And the more biodiversity, the less risk of disease transmission we have. There is this theory called the dilution effect and sink population. When you have so much a diversity, let me say richness as well as abundance of animals in a place, there are some species that will not be viable in transmitting these diseases. So if the disease jumps from one species to another, that another species, which becomes a dead end host, will just prevent the transmission chain. But if we disturb the environment and we have just few species, anything that comes will really the system will really be porous, and there will be high risk of jumping of disease from one point to another, especially from these animals to humans. So the best means where conservation fits into the One Health is that we keep our environment healthy, we avoid hunting our biodiversity, so that there will be this balance. The more animals we have, the less risk of disease transmission we have. All right. So my research on arbovirus stems from the desire to link wildlife population and what affects the change in wildlife population. All right. Um, and the behavior of animals that also affect the population and distribution. Then disease becomes one aspect when I consider studying what affects distribution and population of animals. So we decided to um, carry out this project, which is actually a surveillance project, and it has been going on for about 4 years now, to look for viruses in wildlife, that is in birds especially, so that if we are able to detect viruses in these birds, we can make recommendations for public health action. I mean, that will eventually lead to um, um, response, right? Will eventually lead to early response, early warning detection, and then ultimately public health action that will prevent the transmission of these viruses from the animals to human population. But we know that um, over 70% of emerging diseases are zoonoses. um, I think this slide should summarize what I have been saying all this time. Zoonosis, which means diseases that are transmitted from animals to humans, and sometimes there could be that backward transmission also from humans to animal, anthropozoonosis and all of that. But we know that over 70% of emerging diseases are zoonoses, which means that they jump from animals to humans. And and this actually tells us that the animal population biodiversity houses a lot of diseases. But then I have come to think of it this way, not to implicate our biodiversity, that they have a lot of diseases, and that is why they are jumping from them to animals, I mean to humans. But how about if you look at it this way? Diseases are a lot in nature, but animals are helping to resolve these diseases. So until we go there to the pool of these viruses, we touch the animals, we touch their habitats. That is when transmission is facilitated and we contract this. So for me, I think that um, biodiversity is helping to curtail disease transmission. I read in a place that one thought that says if there are no biodiversity, there will be no disease reservoirs, and there will be no jumping from animal, I mean, to humans. But if you look at it critically, if there is no disease reservoir, um, disease can actually quickly jump or can be transmissible between the human population, which in that case will create more transmission, maybe like uh, an environment that will make transmission so easily um, achievable. For example, if you take a look at the coronavirus that came into human population, humans are social, really social. So if a disease is transmissible only between humans, it's really fatal, because before you know, these humans has contacted it from the other from the other human, from the other human, and from the other human. But if we have a reservoir of these diseases in biodiversity, it is only when we trigger it that they come up and they infect humans. So I believe that biodiversity is playing a very major role in preventing spread of diseases, as we know. All right. So a chunk of so let me continue in the line we started now. So we know that as over 70% of emerging diseases that are emerging and re-emerging every time, they are coming from animal. But then to narrow down my speech, it's about arboviruses. Most of these diseases are actually transmitted by arthropods. All right. They are transmitted by arthropods. Some of them insects, some of them arachnids, and all of that. So my focus actually is arthropod-borne viruses, which is arbovirus. So many arboviruses have actually been linked to birds. I've been talking so widely concerning the biodiversity. Let me narrow it down to birds. The two major highlights now are arbovirus and birds. Okay, so the majority of emerging diseases are zoonoses, and a major portion of these zoonoses are arthropod borne. And many of these arthropod borne diseases are linked to birds. So I hope that makes it very clear now. Um, for example, we have viruses like West Nile virus, Usutu virus, Simbis virus, and St. Louis encephalitis virus, and many more. Some of them are actually not, birds do not serve as a reservoir, but birds can serve as a transporter of the vectors. For example, this CCHF, which is Crimean Congo hemorrhagic fever, is not known to be to infect birds, but it is transmitted by ticks, and ticks are found on birds. So, and as the birds move from place to place, they transport these ticks, and these ticks get established in a place and creating a new foci of disease transmission. So that's why we are interested in birds and the vectors that they carry about. All right. So, but what we don't know is that many of these diseases, these viruses, these arboviruses occur uh, in the backdrop of key knowledge gaps. Okay. So, how the transmission is going on, what is the transmission dynamics, what exactly is causing the jump from humans to animals, I mean, from animals to humans, and all of that. In most cases, we do not know. But if we know, um, outbreak response will really be we will be eased. All right, we will not struggle so much to prevent pandemic and all of that. So it is in that road that we want to understand what are the roles, I mean, the roles of birds in transmitting arboviruses, especially in our axis of the world, in Africa and in West Africa and even in Nigeria, for example. There are a lot of gaps in understanding the ecology of these diseases, especially arboviruses. And so we really need to know that. Okay. So in this work, I will be showing you what we have been doing for the past 4 years, and I will be specific. I will bring up the matter of one virus, which is Simbis virus. Simbis virus is a Togoviridae. I mean, it's in the Togoviridae family. It's an alpha virus that has a bird as its reservoir host, has a mosquito as its vector, and it has humans as the dead end host. I would say that um, it really affects the birds really don't come down. It's not been shown that birds really come down with this virus, but it can cause neurological disorder in humans and a lot of some, rashes and all of those kind of um, symptoms. So in Nigeria, Simbis virus is really not known. Before our work, there was no mention of Simbis virus. I wouldn't say there was no mention of Simbis virus. There was a mention of Simbis virus in 1978 by one Fagbami that did serology and detected the antibody of the virus in humans. But apart from that time, as at that time, there has not been report of Simbis virus. And um, we think that this virus is actually in the population, and in the era where arthropod-borne viruses are actually emerging, jumping from place to place and causing pandemics and epidemics, we really need to understand what is in our environment so that we can prepare against any of such occurrences. So in Nigeria and West Africa, we the knowledge of SIMBIS is actually scarce. So that is why we wanted to know what the role of birds and the associated vector in the transmission and maintenance of this arbovirus, and what the risk stands for humans. Okay, so we the objective was actually to highlight the epidemiology of SIMBIS, like I have said before, because before now we actually did not know which birds were serving as reservoir host. We didn't know the host range, like how many bird species, can be infected by this virus. We don't even know what mosquito species are involved in transmitting this virus. Although, I mean, globally, we know there's a general knowledge that mosquitoes in the um, um, Culex genera can serve as vectors, but we don't know how many of these mosquito species can serve as vectors, and we live with these mosquitoes every day. So they bite the birds, bite us, and then this virus keeps on transmitting around. I mean, so we wanted to detect and characterize the virus in the wild bird population. And usually one problem with research is that they are usually disjointed. One of the reasons, one of the gaps we wanted to cover also was to detect the virus simultaneously in the vector and in the host. And if possible, to also check humans so that we can see the chain of transmission and what dynamics is playing out. And relating that to environmental variables and changes, we'll be able to understand the dynamics and the ecology of this virus. So we started collecting data from 2022, and the main study area was A priori. Can somebody hear me? It's as if I'm talking too much to myself.
We can hear you.
Oh,
Uh,
great.
we are
Yes,
following.
please.
We are following.
Okay.
Yeah, you're not
We
alone.
can
We
hear
can
you,
hear you.
Dickson.
It's just that you
I
can't see
think
us.
Okay. Thank you so much.
we are with you, Dickson.
All right. So, so it was we started sampling in Aplori, that is in Amorum Forest Reserve, and the environment around Amorum Forest Reserve. Aplori is fast becoming a greenhouse. I mean, sorry, a green island in Jos. You know, recently you see people building houses around and all of that. So in the nearest future, you will just see, like you are seeing on this map, houses surrounding the place, and then you just see one green dot at the center. That is actually the future that we are foreseeing. And if that will happen, we need to know what is in this environment. We need to know the risk of things happening so that we will be able to plan ahead. All right. So we collected birds, ring the birds, and then collected ticks from the birds. We also collected plasma samples. We collected blood samples. I mean, very little quantity because we really needed to follow ethical protocol. If you want to collect a sample from a bird, you have to be approved to do that, and you must not collect sample, I mean, especially blood, more than a specific quantity. So we followed all that protocol. And yes, so after that, we took our samples the to the lab, harvested a serum, and stored that for further analysis. We did what this work is actually focusing on is the blood analysis, which is the dry blood spot. We collected the blood on a dry strip and then stored it for RNA extraction to detect the virus. So this is the image of how the samples were stored. This is A priori lab where the samples are stored in -80 freezer so that it can preserve the integrity of the virus. So we also, and then came back all the way to Europe to do the molecular work. Eluted this dry blood spot and extracted RNA. We carried out a real-time, I mean, quantitative PCR And we also sequenced the positive samples and using twist target enrichment and amplicon based sequencing. It was a lot of work. Um, Sometimes these technologies really don't give you what you want. So we, for the twist target enrichment, we did that, but we didn't get a full coverage of the virus genome. So we decided to do amplicon based, where we have to generate a sequence of primers that covers the genome of the virus. Then we'll begin to amplify them one after the other, and then came back to assemble them for us to be able to get the full genome. So for those, I know this is not a very familiar presentation, but I just brought some details so that at least we know what's happening. So this machine is a qPCR machine where you amplify the genetic material of the virus. And here below here, the graph here is the one that shows it's a real-time amplification. So as it's amplifying, you are seeing how it's amplifying, and to a particular threshold, you see how it goes until the amplification and the time that you said is elapses, and the amplification goes. But overall, we see that the amplification curve starts from around 0 to 40 CT values. That CT value means cycle threshold. After that threshold, any amplification will not be seen because it's the material. Even if there is any material, it will not be amplified, and we cannot confirm that it's positive. Okay, so the lower the threshold, that is the lower the threshold, the higher concentration of the genetic material we have. So far we sampled several species, but also there was some soft spots for African thrush, since we know that African thrush was this kind of species that is everywhere, dirty in a way, it doesn't discriminate to go somewhere or to eat something and all of that. So it's a very good species to study what birds pick from the environment and how that can affect subsequent transmission to humans. All right. So so majority of our samples were African thrushes, and surprisingly, it was only African thrush that were positive for Simbis viruses. So three of the African thrushes were positive via um, quantitative reverse transcriptive PCR. And here you can see the African thrush is there. So out of 504 samples, we actually sampled 504 blood samples. 3%, I mean 3, which is like 0.06, was positive, and it was African thrush. Um, I know that this is actually a very low number, but for me, when you talk about disease, prevalence can really be overrated. All right. Um, because if you are looking at prevalence solely as your yardstick um, for judging the risk of disease, uh, sometimes we may be misinformed, because some diseases are highly and highly potent. And so if you detect one in a population, it means that there is high risk. And yeah, so I think that that is what actually is playing out in this situation. We have very low number that were positive. You can imagine out of 504 samples, you have just three that were positive um, for RNA of Simbis virus. But then there is something I need you to know, that we just have very low positive samples does not mean the disease is not in circulation. It also has to do with the ecology of the virus. Okay. So, for example, this virus is only active in the bird for just like 3 to 4 days. So, the active viremic period is just like 3 to 4 days. So, after that, it will be difficult to get the virus in the bird. So, the only thing you can use to trace that this bird has been infected is antibody presence. So you get the antibody, and then it gives you the history of infection. So we have three samples that were positive uh, because of the difficulty in catching the virus red-handed in the birds. Because if you sample the birds this week and you leave like 3 4 days, there is high likelihood that the birds becomes positive, and you know that they usually don't come down with this virus. So in the next week, in the next 5 days, you come and sample that bird again, you will not see that virus again. So it is as tricky as that. So we are even glad that we were able to even track down like three birds that were actively positive for this virus. Okay, so what actually was very interesting to us was the fact that this virus, when we constructed the phylogenetic relationship using a maximum likelihood method, we found out that the virus was the one we found in Nigeria was closely related to the one detected in mosquito in Algeria and in Kenya, as well as in um, Spain. So one of our thoughts that we had in mind was to actually track the movement of this virus and to implicate migratory birds. Perhaps that as migratory birds come from Europe, and then before they go and all of that, this movement around, they can introduce this virus to our place because we have not recorded this virus in birds I mean, it has really not been recorded in birds in Nigeria before. So the first thing you would think is that, okay, a Nigerian birds population do not have this virus. So it's likely that these birds are coming from Europe where the virus is endemic. You know, for example, places like the Netherlands and the Finland and Sweden, they actually this virus is endemic in these places, and birds come from those places to Nigeria. So the first thing you would think about is that, okay, maybe it's these birds that brought these viruses. But then the phylogeny is telling us that this virus is not coming from Europe, it's coming from within Africa. And this is actually pointing us to the fact that this virus is in circulation, but then it has not been detected. So as our birds move around in Africa, intra-African migrants like the cuckoos and even the African thrush that is suspected to do some little movement. So they carry these viruses around. Okay. So the point I'm trying to make here is that we have this virus circulating in our bird population, and it is indigenous. I mean, well, not indigenous. It is enzootic that is present in our birds population, and we should not implicate some migratory birds coming to introduce the virus to us. Okay. By the way, the Simbis virus was introduced from Africa to Europe before it became endemic in that place. So this is one key result that we found, and this is very, very important. And this points to the fact that if we monitor our biodiversity, we can actually detect what is in them and what is in the environment, uh, so that it can inform public health intervention. It can even inform preparedness, so that if anything happens, we will know that, okay, we have a checklist of things, of viruses in our environment. So let's not look so far away. And given the fact that our system is not so strong, our diagnostics is not really really strong. It's possible that some of these viruses are in the population and they are misdiagnosed. So it's better if we do surveillance and we detect this population and we detect these viruses, we'll be able to, it can also ease diagnosis. Okay. Some of these viruses have similar symptoms. So you may be diagnosing one virus as another virus. So there's that chance of misdiagnosis. But if we know that this virus is in this population, then we we can make headway, and we can prepare ourselves against a pandemic and outbreak. So for to bring it back to us, this is how this is Spain up there, and that is Algeria, this is Nigeria, and then Kenya is there. The thing is, the major thing, the surprising thing to me personally is how this virus is closely related to the Kenyan virus, Kenyan strain. Kenya, far East Africa, Nigeria, West Africa there, and but you know that the birds actually do not have border as well, so they move around. Okay, so they move around and they introduce these viruses from one point to another. So we have published this paper, and in case you want to take a look at the main paper to understand exactly what we have done, but I hope I've been able to break down some of the things we have done to you. Then this brings to the last aspect of this. What does all this mean? What does it mean? I think I have said a little about this. Um, Now we know for the very first time that the birds in Nigeria, and especially in Plateau, carry Simbi's virus. And now that the place is becoming seriously urbanized, there is this risk of transmission of this virus from these animals to humans. Another thing is that the mosquito population, you know, for diseases that have vectors, if you detect the disease in an animal, either in an animal host or a human host, you surely know that it's not coming from anywhere, that the vector of that disease is in that environment. And that is what has really been lacking when I made a statement initially that most of our researches are disjointed. Somebody will go and sample mosquito from somewhere and just do the entomology, and sometimes we do the virology, and that will be all. Somebody also does will go and sample birds from somewhere and just does the virology and all. But then we want a situation where we can connect all these things. All right, we can connect them all at once in the same environment. The birds are there, the mosquitoes are there, the humans are there. The enabling environment, water body, temperature, optimum temperature for transmission, they are all there. So in this case, we are able to reveal the underlying factors that can enable this the transmission of these diseases. And something I didn't mention actually is that most of these viruses were detected in the dry season. And in the dry season, we have streams, streams that are usually reduced to pools. When streams are reduced to pools, you see that birds go there to feed, to drink during the dry season and hot heat season. They go there to cool off. The mosquitoes there, even though they may not be as abundant as in the wet season, mosquitoes are also there breeding and all of that. So that is where they keep. I mean, they hibernate. I wouldn't say hibernate, but that is where the population survives until the next rainy season, where the population explodes again. So that interface, studying that interface and what is happening around there, can give us an indication of the factors that are playing part in the transmission of these diseases. So by and large, we know that we have to create a system of surveillance where we can monitor our biodiversity, knowing that in as much as we are conserving the place, we should also monitor their health. It is, I mean, it's fortunate for us that some of these diseases are not killing our birds. Even though I know that sometimes you see birds die of or something, you just be thinking, oh, what exactly is killing this bird? Is it that farmers have poisoned the birds? Obviously, sometimes there is a vehicle strike, and then some of them you find them dead on the way. But then in Europe, you will see that a whole population of bird is wiped off by a virus, especially Usutu virus, which is a notorious virus for the blackbirds in Europe. So I'm wondering why. I mean, I'm thankful that we don't have such situation in our place yet. But then, now that we know that these things are actually existing in our place, we have an upper hand. We can prepare when outbreak occurs. We can also um, prepare to, or we can it's a way to also detect, an early detection of this virus is happening, I mean, going on in our environment. For example, I consider that this work that we have done, it can be categorized into early detection, all right, where we know there has not been I mean, there has not been pandemic or epidemics of this virus in Nigeria yet. But now our work has exposed the virus. Now we have detected it very early, even though it may have been existing for a long time in the population. But for now, we know that, okay, it's early enough because there has not been pandemics, I mean, outbreak. So now we have the knowledge that this virus in the population, and we'll be able to prepare against this. Okay. So, and um, where the whole One Health thing comes in is the fact that, okay, me, originally an ecologist, is doing this work, detecting this work. I present this work and I give it, people in the health sector, the medical doctors and even the vets, they they come to an understanding that, okay, this actually is in the world, is in the population. And with that, it circulates information to be able to detect the virus, diagnose the virus, and then prevent future outbreaks. So I hope this has been interesting to you. So I think I should end the talk here. So far, so good. I hope I've been able to convince you and not confuse you. Thank you very much.